Yo, 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 this is Mr. Tran with another video. I'm going to be covering experimental design. Some of y'all asked, what is an experiment, Mr. Tran? And I'll be answering that today. An experiment is a process that is reproducible by which an idea is tested. An experiment allows for three things. Manipulation of variables of interest. This is our independent variable, but we'll get to that. Hold constant control of other variables, and doing the experiment again and getting the same results. We're going to go over the parts of an experiment. This is super crucial, y'all. When we think about an experiment, we have to think about groups and variables. So let's go over the groups first. First off, we have our control group. The control group is used for comparison to other group. It's like saying it's the baseline, okay? And then our second group is the experimental group. The key difference between the experimental group and the control group is that the experimental group must differ from the control by one variable. And when we talk about these groups, y'all, we need to have replicates. So at least 3 replicates are needed per group. That means for a good experiment, there needs to be 3 replicates in a control group and 3 replicates in an experimental group. Next off, we have our variables. So first, we have the independent variable. This is the variable that you change between groups. It's the one thing you can control, okay? And then our second variable is the dependent variable. The dependent variable is the thing that changes as a result, and you can measure this. The dependent you can measure, and recapping, independent you can change. You yourself, the scientist, the student, can change that variable. And last but not least, we have the controlled variables. Sound like a controlled group, but we're talking about controlled variable here. The controlled variable is factors or are factors that are kept constant so they don't affect the results. So when I know a good experiment is being conducted is when the only thing that changes is one variable, that independent variable. So I should have everything else constant. There are 2 things that are needed for a good experiment. And again, 1, good replication, being able to repeat the experiment. And number 2, identifying all the controlled variables. This means we eliminate something called the confounding variable. In the late 1500s, a scientist named Van Helmont wondered, how do plants grow? He hypothesized that plants eat dirt. So Van Helmont decided to grow a tree and test his hypothesis out. He carefully masked the amount of dirt, and then he masked the sampling as well. For a year, he carefully watered the plant and the tree grew. At the end of the year, Van Helmont was ready to check his hypothesis. He dug up the tree and masked it. He carefully masked the dirt as well. The dirt was only less by 1 kilogram. What? While disappointed, Van Helmont did come to a conclusion. He concluded that plants do not eat dirt. Instead, plants grow by drinking water. Y'all, there's something wrong with that conclusion. Can y'all think of it? You guessed correctly. You cannot conclude something that you were not testing in your experiment. The idea that plants eat dirt is simply rejected by this experiment. There were other things wrong with this experiment, y'all. There was no control group to compare. Many variables were being manipulated here. The amount of sunlight, water, he didn't measure any of that. He didn't repeat the experiment to see if he would get the same results as well. And he didn't have sample size. So, gonna stop bashing Van Helmont now because I was in the 1500s. Let's move forward in time. In 1668, an Italian scientist, Franchesco Redi, was not convinced that life came from non-living things. Let's put 1668 into perspective. In American history, 1668 was when Massachusetts annexed Maine. Y'all, states were already made, and we still believed in this. Let's expand on what this idea is. People used to think that living things contained a vital spark, something that made things alive. When living things died, this vital spark could be transferred to new organisms. For example, left alone in a corner, rags made of wool would spontaneously generate mice. The reason for mice and not sheep is that the wool had only enough spark to generate smaller organisms, a mouse. In the same way, meat, being dead and cut up, could only generate flies from its vital spark, and not a new sheep or pig or cow or fish. Y'all, wow. Just wow. So Redi didn't believe in this. He believed that like comes from like. In other words, adult flies produce new flies. Unfortunately, no one has seen a baby fly. So as a control group, he set an uncovered jar, he put a dead fish in, flies would go and be able to land on the fish, and then the result of this control group is that maggots appeared. So Redi had an experimental condition as well. The experimental condition was that he put the fish in, he covered the jar with glass. Flies couldn't land on it because the glass was blocking the flies. And the result of this experiment was that no maggots appeared. But no air was in the jar either. So there were two variables being manipulated here, no air as well as no flies. So he couldn't correctly conclude which part contributed to the no maggots. So he had to design another condition. Redi's second experimental condition was that he covered the jar with a cheesecloth after inserting the fish in. Flies couldn't land on the fish, and the result was that there were no maggots, and also the air was allowed inside the jar because the cheesecloth was porous enough to allow air to go in, but big enough to prevent flies from landing on the fish inside. This condition was a good condition to compare the control group. So let's compare between Van Helmont and Redi. Redi had a group to compare his experimental results to, while Van Helmont did not. Redi had only a single variable tested. Redi's experiment, however, lacked adequate multiple trials or multiple setups. Remember, experiment must be repeated in order to have the results be trusted. Spontaneous generation was what Redi was trying to disprove. Louis Pasteur actually managed to discredit the theory for good. Pasteur took several swan-necked flasks, filled them with broth from a hay infusion, and then boiled the broth in the flask. Boiling killed all living things in the flask. The design of the flask allowed air in, but kept microorganisms out. Skeptics insisted that the boiling had killed the vital spark, so Pasteur tilted some of the flask on their side. This allowed some of the trapped microorganisms into the infusion of the broth. The broth rotted and became really, really stinky. Only flasks tipped or with their necks broken got smelling. Some of the upright flasks remained microorganism free for an extended period of time. For example, from 1864 to 1866. That's nearly 2 years, y'all. The reason why some of these actually expired was because of the endospores. The endospores are just dormant and highly resistant cells bacteria produce in order to preserve the genetic material. Truly sterile broth would remain clear until it evaporated. In the end, Louis Pasteur did indeed demonstrate that life doesn't spontaneously appear from this concept of vital spark. So Louis Pasteur did demonstrate good scientific design. So again, he tested only one variable. He clearly identified what is being tested. Those are identifying your independent, dependent, and then try to eliminate confounding variables by keeping everything in the experiment constant except for the one independent variable. He also had multiple trials and at least 3 sample size for each of those groups, the flask that was upright and the flask that was tilted. So in the experiment, it mentioned many flasks. So again, those are essential parts of a good science experiment, y'all. Hopefully y'all took notes, learned something, and y'all have a good one.